Hello and welcome to my channel. This lecture will continue our discussion of the nervous system, so let's begin. Today's lecture is about the meninges. I think we studied this before in the head and neck lectures in anatomy, but today it's going to be a revision on the meninges of the nervous system. The meninges are three layers from outside inwards: the dura mater and arachnoid mater, and then the pia maters. The dura and arachnoid, they do not go with the sulci and gyri of the brain, but the pia mater is adherent to the brain tissue and goes into the sulci and gyri. Remember that. We've seen what happens to the meninges in the spinal cord, and today we're going to discuss the organization inside the skull. The dura mater has two layers. Outer layer called the periosteal layer. Actually, it's the periosteum of the cranium of the skull. And called endosteum or endocranium. It's continuous through the foramina with the periosteum on the outside of the skull. The meningeal layer, which form folds, as you can see here, it's close contact with the arachnoid and continues in the spinal cord as the spinal dura through the foramen magnum. The two layers separate to form the dural partitions and venous channels. Dural partitions, formed by the folds of the dura. There are two vertical and two horizontal folds. The two vertical folds are the falx cerebri, this one, falx cerebelli, this one, and Tentorium cerebelli, this is horizontal fold, and the other horizontal fold is the one that covers the pituitary gland. It's called the diaphragma cilia. So it's falx cerebri, falx cerebelli, Tentorium cerebelli, and diaphragma cilia, two vertical and two horizontal folds. The falx cerebri, which is the largest of the folds, it's a vertical fold between the cerebral hemispheres. It means that it occupies the interhemispheric fissure, the fissure between the two cerebral hemispheres, to right above the corpus callosum, and it attaches anteriorly, as you can see here, to a bone protrusion called the crista galli, and posteriorly crista galli and frontal crest. Posteriorly, it's attached to the top of the tentorium cerebelli at the internal occipital protuberance, as you can see here. It has a free margin in which runs the inferior sagittal sinus, the sinus you see here. Okay, these are the attachments of the falx cerebri. It prevents rotation of the brain. It occupies the interhemispheric fissure, so it prevents rotation of the brain around its axis. The tentorium cerebelli is a horizontal fold. This one, see? A horizontal fold that covers the cerebellum, and it's attached posteriorly to the occipital bone along a groove for the transverse sinus. This is the transverse sinus, the one you see here. Attached laterally to superior border of the petrostemporal bone. This is the petrostemporal bone. Okay, it attaches to its superior border, and ends anteriorly at anterior and posterior clinoid processes. These are the clinoid processes. These are the posterior clinoid processes. and these are the anterior clinoid processes, attaches to both, and it has a free margin anteriorly for the passage of the brainstem. Okay, this is called the tentorial notch for the passage of the brain stem. The falx cerebelli, another vertical fold. It's small and attaches posterior to the internal occipital protuberance or crest, and superiorly to the tentorium cerebelli, as you can see here. This is the end of the tentorium, the top of the tentorium cerebelli, and this is the falx cerebelli. And it has a free anterior edge between the two cerebellar hemispheres. So the falx cerebri between the two cerebral hemispheres, and the falx cerebelli between the two cerebellar hemispheres. Another horizontal fold called the diaphragma celli means the one that covers the sella turcica, the site for the pituitary gland, or hypophysis cerebri. It's a small horizontal shelf covering hypophyseal fossa in the sella turcica of the sphenoid bone, as you can see here, and it has a central opening, of course, for the passage of the pituitary stalk, the connection between the pituitary and the hypothalamus above it, through the diaphragma celli. Has a central opening for the infundibulum of the pituitary gland to pass through the diaphragm. So this is the diaphragma celli. Arterial supply of the dura. The arteries that supply the dura run in the outer periosteal layer. It's the anterior meningeal, middle meningeal, and posterior meningeal and accessory meningeal arteries. So rich in blood supply, and these are the arteries, as you can see here. The branches of the arteries, they leave an impression on the inside of the skull. The middle meningeal artery supplies the greatest part of the dura, and it's the one involved in injuries, especially at the pterion. The pterion, we studied in the head and neck. It's part of the skull where the frontal, sphenoid, and parietal bones meet at the front. It's almost at this region, okay, where the middle meningeal artery divides, as you can see here. This is the divisions of the middle meningeal artery. Anterior meningeal arteries, branches are of anterior ethmoidal arteries, and posterior meningeal artery is the terminal branch of the ascending pharyngeal. We studied this with the branches of external carotid artery in the neck, if you remember. The ascending pharyngeal enters the cranial cavity through the jugular foramen and continues as the posterior meningeal, and it has a meningeal branch also that passes through the hypoglossal canal, also for the supply of dura. Accessory meningeal artery, it's from maxillary artery. We studied this with the maxillary artery branches in the infratemporal fossa, and it enters the cranial cavity through the foramen ovale. So remember, the middle meningeal supplies the greatest part of the dura. Anterior meningeal artery is a branch of the anterior ethmoidal artery. Posterior meningeal is the continuation of the ascending pharyngeal, which is a branch of the external carotid and passes through the jugular foramen, and has a meningeal branch also that passes through the hypoglossal canal. And the accessory meningeal is a branch from the maxillary artery in the infratemporal fossa that enters the cranial cavity through the foramen ovale, together with the mandibular nerve, if you remember. Okay, arachnoid mater is the thin avascular membrane, lines but is not adherent to the dura mater. It's the one you can see here. This in orange. This is the arachnoid mater, and the gray one is the dura mater, the outer layer and inner layers of the dura mater. This is the arachnoid, forms here the arachnoid granulations. It has thin processes on the inner surface. See these processes that crosses the subarachnoid space and continues with the Pia mater. The Pia mater is the other orange layer here directly on the surface of the brain. So this is the subarachnoid space. It does not enter the grooves or the fissures except for the longitudinal fissure. Okay, so only one fissure it goes through, the longitudinal fissure. Of course, it goes through it because it covers the surface of the brain. The Pia mater is the epineurium actually of the brain. It's a thin, delicate membrane adherent to the brain tissue and invests the brain. Line the grooves and fissures and goes with the sulci and gyri and closely applied to roots of cranial nerves. So we have the arachnoid and the Pia maters, and now you can distinguish between them. Remember that the Pia mater is the only one that goes into the sulci and gyri of the brain. Okay, spaces. We have extradural space between the outer layer and bone, between the outer layer of the dura and bone, and filled by blood if there is injury or hemorrhage from the middle meningeal artery. So, if you suspect a brain injury or intracranial hemorrhage and you performed a CT scan or an MRI on the patient and you find a collection of blood outside the brain between the membrane and the bone, this is epidural hemorrhage or extradural hemorrhage, and the source of which is the middle meningeal artery. And because the space is tight, middle meningeal artery is pushed against the bone, actually it grooves the bone, so it takes a while for the bleeding to occur. That's why you have to confine the patient under observation for 24 hours in a hospital after a suspected brain injury, especially if he lost consciousness during that injury, because the symptoms are delayed. They occur in the next day or the day after. Okay, this is the extradural space. The subdural space, it's between the dura and the arachnoid. It's a potential space. It's filled with blood in cerebral veins injury. Which cerebral veins? The superolateral cerebral veins. These are the superolateral cerebral veins here. As you can see, they drain the superolateral surface of the brain and drain into the superior sagittal sinus. And because they drain into the superior sagittal sinus, they have to pass through the arachnoid and the space beneath the dura, which is the subdural space, and then the dura, then to the superior sagittal sinus. And this occurs actually in old people, more common in old people when they hit their heads, and it's delayed symptoms in which, if a patient bangs his head against the sink, for example, and loses consciousness, the symptoms may appear a week or two later because collection of blood in the subdural space. So if you find a collection in the subdural space, the source of which is the superolateral cerebral veins. In the subarachnoid space, between the arachnoid and Pia, of course it's filled with the CSF, cerebrospinal fluid, and the space continues around the spinal cord. It enlarges to form cisterna. We studied the cisterna of the CSF in the last lecture when we discussed the circulation and production of the CSF. The space contains CSF and blood vessels. The blood vessels of the brain, they run in the subarachnoid space. Remember that. And the choroid plexus in the ventricles are responsible for the production. They are called tila choroidi, the choroid plexus or tila choroidi. They produce the CSF. Remember, for the production of CSF is the choroid plexus or the tila choroidi, and for drainage of CSF is the arachnoid villi. Arachnoid villi drain the CSF into venous blood, and they project as clumps, as you can see here, called the arachnoid granulations, that projects into the nearby sinuses, and especially the superior sagittal sinus and its lateral lacunae. There are lacunae of blood lateral to the superior sagittal sinus, as you can see here. Okay, this was a brief idea about the meninges of the brain. I hope you got the maximum benefit from this lecture, and thank you very much for watching.